genetics and immunity. In this last segment, we are going to address some abnormalities associated with our immune system. And these abnormalities can be caused by mutations or can be acquired. Now, our immune system continually adapts to our environment because our environment is changing. So we adapt to these changes. Our immune system is going to be able to break down particles that come in us. However, when this immune system is affected, they are not going to be able to properly address these infectious particles that come in us. So, in that case, the condition can be induced by some kind of acquired infection or acquired immunodeficiency. Sometimes it can be inherited because a mutation occurred in the gametes, right? And now they are passed down to offsprings. So, because of this modification that can occur, our immune system can be sometimes too weak, sometimes too strong, sometimes the immune system can be completely misdirected. What does that mean? That means that, well, instead of doing what is intended to do, it's going to do something else, right? And these abnormalities can be caused by multiple genes that are somehow modified or interacting in an inappropriate manner. Sometimes it can be because of mutations in a single gene. And regardless if it's polygenic or it occurs because of a single gene, the outcome is the same. It's that our immune system is misbehaving, right? To this day, there are about 20 different types of abnormalities in our immune system that are associated with our genetic background. Now, as I say this immunity issues are associated with both innate immunity but also adaptive immunity. Now, there are some conditions including the chronic granulomatous disease, which is caused by a mutation in a gene that encodes an enzyme that is called an oxidase. Now, that oxidase, when it is mutated, some white blood cells, more specifically the neutrophils, right, which job is to engulf foreign particles, including bacteria, and they are supposed to break down this bacteria after they engulf them. Well, because of the lack of that enzyme, these neutrophils are not going to be able to destroy the bacteria, which is actually a problem, right? Another condition that I would like to mention is the severe combined immune deficiencies, which is actually a group of conditions, right, that we also call SCIDs, right? The SCIDs are going to impact both antibody-mediated immunity and cellular-mediated immunity, right? And this is going to occur because of the lack in the maturation of B cells that are involved in antibody-mediated immunity and T cells that are involved in cell-mediated immunity, right? So there are cases, as you can see, for instance, on this slide, this little boy right, was born with a defect, and that defect is associated with an X-linked recessive allele, right? That is causing this child to have SCID. So this child was forced to live in a bubble because he was born without a thymus. And you know that the thymus role is to allow our white blood cells to learn the difference between self and non-self. With the T cells not being able to learn the difference between self and non-self, well, this child is completely unable to mount an immunity that protects him against infectious particles, including bacteria and viruses. So he becomes so vulnerable, and anything can cause this child to be sick. So as a consequence of his genetic condition, he could not have activation of B cells or even maturation of B cells, which actually caused him to, live a completely isolated life. There are many inherited immune deficiencies that we know of these days. I already mentioned the chronic granulomatous disease. All right, there are some conditions in which the defects affect the thymus in a sense that there is no thymus. If there is no thymus, well, there is no T cells, right? Okay. There are also other conditions associated with neutrophil immunodeficiency syndromes. This is caused by T cells and B cells and neutrophils that are not able to do their job. Now, here is what I'm not going to bug you with. Let me tell you that up front. I'm not going to ask you to know specifically which gene is responsible for these conditions. I do not expect you to know this, right? But I want you to be somewhat familiar with some of these conditions, right? The SCIDs are groups of immune issues, right? Including adenosine deaminase deficiency. Deaminase means that there is an enzyme which is supposed to do deamination, so take away the amino group, right, from an amino acid, right? That enzyme is not there. As consequence of that, well, you have no T cells, no B cells. We have adenosine deaminase deficiency with sensitivity to ionizing radiations. In this condition, there is no T, no B, or natural killers that are going to be there functional, right? So let me only mention this group of inherited immunodeficiency diseases. Now, let's dive into HIV, the human immunodeficiency virus. How is HIV affecting our immune system? This is what we are going to address now. So, HIV occurs through direct contact. What do we mean by direct contact? Body fluid from one person who is infected to another person who is infected in terms of body fluid, and this is at a blood level, right? And sexual intercourse, right? So, HIV viruses are able to infect macrophages and helper T cells. How are they able to do so? By attaching to surface molecules on these cells. These surface molecules are CD4 and CCR5. Now, CCR5 is what we call a co-receptor. In fact, CCR5 does not work alone, right? It's working alongside with CD4, right? So, as the virus attaches to our cells, they get in, and when they get in, they are able to multiply, right? They are able to multiply, and as they multiply, ultimately, they are going to cause our cells to burst and die. And when our helper T cells die, and you know the role of helper T cells, which is to regulate our immune response, to stimulate the maturation of B cells, the multiplication of T cells, right? Clonal expansion cannot occur without helper T cells, right? So with losing a large amount of helper T cells, we definitely become immunocompromised, right? So the less of helper T cell is going to basically prevent B cells from being activated, from undergoing clonal expansion, right? Converting B cells to plasma cells that actively produce what? Antibodies, right? And release those antibodies. But let me close that parenthesis. So now, infections are going to occur because of the lack of helper T cells and therefore B cells, right? So these viruses are going to replicate very fast, okay? Now, here is another thing that is worth mentioning and very important to know. It's that these cells are, not cells, these viruses have also this bad habit that they are going to mutate easily, and they can also hide. Well, where do they hide? They are hiding within our own cells, within the host cells. All right, so here we have HIV. So here we do have an HIV virus, okay? We do have a HIV virus here with some surface markers, including a protein called GP41 and another protein called GP120. Now, GP120 is essential for the attachment of HIV on our cells. Very important to remember. Now, when we look at our cells, so here we have our cells, right? We have two markers on our cells. One is the CD4 receptor, okay? So that is the proteins on which GP120 is going to attach. And now here is another one, CCR5 receptor or co-receptor. That CCR5 is going to help the attachment of the HIV virus on our own cells. So here we do have an interaction between HIV virus and our cells. Notice that HIV, it's an enveloped virus. So we're going to have a phospholipid bilayer right here, right? That's a phospholipid bilayer. And we also do have a phospholipid bilayer, but let me close that parenthesis. Now, let's take a look at the cycle of infection of HIV when it comes in our cell. So, when HIV comes in our system, what specifically happened? So first and foremost, the virus is going to bind on our cells, right? GP120, which is a protein that is part of the envelope of the HIV virus, is going to attach to our receptor, specifically the CD4s and CCR5 co-receptors. So they attach, and then the membrane of the virus and our membranes are going to merge. And as they merge, what happened is that the capsid, along with the RNA of the virus, are going to migrate toward the inside of our cells. After that, we are going to have a disorganization of the protein coat, so the capsid, right? So that the genetic information, the RNA, the viral RNA, can be laid in our cytoplasm. So at that point, know that the virus, beside coming with its RNA, comes also with an enzyme that is called reverse transcriptase. That reverse transcriptase, RT, is the enzyme that is actually going to catalyze the reaction of converting RNA, the viral RNA, into a complementary strand of DNA. Okay? So now we have a hybrid molecule of RNA and a complementary strand of DNA. Now, eventually, the two strands are going to separate, and our system, seeing a complementary strand of DNA, a piece of DNA, single-stranded molecule, our system is going to think, wait a minute. Well, a single-stranded DNA molecule in the cytoplasm, well, you don't belong there, and you have no business in being single-stranded molecule. So let's convert you into a double-stranded molecule. So your own machinery is going to synthesize a second strand of DNA so that you have a double-stranded DNA molecule. Then, after that, our cells are going to see that double-stranded DNA molecule and say, wait a minute, a double-stranded DNA molecule in the cytoplasm, you don't belong there. You belong to the nucleus. Therefore, the double-stranded DNA molecule is going to be carried into the nucleus. And at that point, you are going to have that viral genetic information sitting in the nucleus and floating around. Our system is going to further think, wait a minute, a small piece of double-stranded DNA molecule sitting around, well, you have to be integrated into a chromosome. As a consequence of that, there is an integration of the viral DNA in our chromosome. Where specifically is that integration is going to happen? Well, we cannot say ahead of time where specifically that integration is going to happen, right? And because that integration can happen about almost anywhere in a chromosome, well, guess what? Some genes can be mutated at that point, right? Can be altered. And the gene expression can also be altered. But let me close that parenthesis. You can now start imagining some of the issues that can happen in the proper functioning of the cell with the insertion of viral DNA in our chromosomes. But let me close that parenthesis. Eventually, because the viral DNA is now part of our chromosome, that viral DNA will be expressed just like our own DNA. So, transcription is going to happen, and then translation is going to happen. Now, here is the interesting thing. Because the viral DNA is much smaller than our genetic information, what ultimately is going to happen is that very quickly we are going to have a bunch of messenger RNA, that is viral messenger RNA, which means that the frequency of expression of the genes of the virus is going to be much higher than the frequency of expression of our own genes. As a consequence of that, viral particles are going to have a bunch of proteins, and eventually the viral particles will start assembling in a capsid just like this, and then an envelope is going to form around the capsid, and these viruses are going to completely assemble and leave the cell. As they leave the cell, and because of the number of the viruses, well, our cells are going to burst and die. So this is the cycle. And once you have this multiple viruses, not one is going to be released, but hundreds of them, right? Now, from one viruses we started with, we are going to have multiple hundreds and hundreds, if not thousands of viruses that are going to now go around and infect other white blood cells and causing our white blood cells to die, and eventually we become immunocompromised. All right, so AIDS, which is the clinical form of HIV infection, arise when our immune system start really falling apart because of so many helper T cells dying, right? So at that point, we are becoming vulnerable to about any infection, right? So AIDS, it's the disease part of HIV infection, because having HIV infection doesn't mean you are sick. Okay, let me repeat that. Having HIV infection doesn't mean you are sick, right? You are sick when the HIV viruses are so many that your immune system is now weak, and now flu can basically cause you to die. Any minor infection can now become a major problem, right? So HIV is going to progress and eventually impair your immune system, and that impairment can be generalized through the body and cause you to become very, very sick. So there are ways to tackle the issue, right? By using antivirals, right? These antivirals can attack many aspects of the virus. It can attack the entry of the virus to prevent the virus from getting inside of our cells. It can prevent the replication of the virus by interfering with the reverse transcriptase, so that the reverse transcriptase is not able to convert the viral RNA into the complementary strand of DNA. That's a possibility. Another possibility, it's going to be to prevent the insertion of the viral DNA into our chromosomes. That's another possibility. The last possibility would be to interfere with the virus assembly, right? That's another possibility. So we can interfere with the entry of the virus into the T cells. We can interfere with the replication of the virus and the processing of the viral proteins, which is interfering with the packaging of the virus. All right. So, what are some drugs that are used? We have some reverse transcriptase inhibitors, right? These ones are going to block the reverse transcriptase enzyme, right? We have the proteases inhibitors. These ones are going to block the shortening of certain viral proteins. We have some drugs that are going to interfere with the fusion, right? These are fusion inhibitors. They are going to block the virus ability to bind to our cells, right? And the entry inhibitors, they block the ability of HIV to enter in our cells. Now, let me tell you a little bit about the CCR5 gene. Now, it has been shown that some individuals in human have a CCR gene that is different from other individuals. As a consequence of that, we see that some people can actually be protected from ever developing HIV infection. Why? Because the CCR5 gene is modified in this individual so that this CCR5 gene is not helping the attachment of HIV on the surface of our T cells. As a consequence of that, they become actually resistant to HIV infection, which is great, right? Which is great. So the individuals that are homozygous for a 32 base deletion in that gene CCR5 are completely resistant to HIV infection. However, those who are heterozygous individuals are going to have a lower chance of being infected, even though they can still be infected. That's one. Two, when they get infected, really, they don't really get sick like most of other people who do not have an altered version of CCR5. All right, so there is a condition that we call immunological amnesia. That condition, immunological amnesia, it's that we get infected, we are able to fight off that infection and withstand that infection. However, over time, our body is behaving as if we never actually encountered that infection before. So that each time we get infected, our body has to start all the protection system from scratch, right? So that is basically what we call immunological amnesia, right? Where our immune system is losing the ability to actually protect itself against an infection that we already encounter at some points, right? Other conditions that are part of abnormalities, it's autoimmunity. Autoimmunity, it's when our immune system is not able to tell apart self from non-self, right? This happens in the population, and when that happens, that can cause, for instance, organ failures such as kidney failure, right? Because our kidneys are being attacked by our own antibodies, right? So typically, this is not going to be caused by one single gene mutation. most of the time it's going to be multiple gene, but it can also be acquired, right? Viral infections can cause these issues, right? So viral infections can cause these issues, as I already mentioned this, right? Some viruses that cause some thymus cells to die, right? This is going to cause this condition to occur. Remember that the thymus is the organ where white blood cells learn to tell apart self from non-self, right? And if this thymus cells are dying, well, that is a problem, okay? Some autoimmune disorders are going to be, for instance, diabetes. Type 1 diabetes is caused by sometimes a viral infection in early childhood can cause that, right? It can be a genetic defect that one was born with, right? Where pancreatic cells are called beta cells that are responsible for producing insulin are not there and not be able to produce insulin. We have the Graves' disease that is caused by a thyroid gland cell that is defective. We have hemolytic anemia. In the case of hemolytic anemia, the rhesus factor is the target of antibodies, which is causing cells to die. Multiple sclerosis, it's another condition where myelin in the white matter of our central immune system is attacked by our immune system. Myasthenia gravis, okay? This is when we have neurotransmitter receptors on the skeletal muscles that are defective. We have the rheumatic fever, which is a condition in which heart valves are affected. Rheumatoid arthritis, systemic lupus erythematosus. So this is a condition of the connective tissue. And then we have the ulcerative colitis. Ulcerative colitis, it's basically when our colon cells start dying because of our own immune system. So these are some examples of autoimmune diseases.